hey, didn't see you there. You're just in time to join me for another exciting new episode of Nature Time. On this adventure, we'll be exploring all the neat things there are to know about microevolution and the Hardy-Weinberg equilibrium. Wow, I got the butterflies in my stomach already. Come on, let's go. Boy. Nick's Nature Time is coming at you live, here in the Outback. I'm here with Dr. Stacy Gorman-Schrugenbach, here to talk about the flounder effect and the bottle cap effect.
Actually, it's a founder and bottleneck effect. You see, the founder effect occurs when a few individuals become isolated from a large population. Those individuals who become separated evolve within each other, with no other contact with their previous populations. Those individuals who become separated evolve within each other, with no other contact with other previous populations. This isolated evolution can create a new population whose gene pools differ from the original large source population. Because of this, new species can develop separately and differently from other populations. This is an example of genetic drift, when chance events alter the way allele frequencies of the population.
Wow.
Australia is a perfect example of this. Long before time, all the continents in the world were one, Pangaea. As time progressed, Pangaea began to separate, eventually causing the isolation of Australia from the other continents. The animals on the island did not have any contact with any other species in the original population. Over time, in many generations, they evolved separately, creating species like no other found in the world.
Oh, that's some real swell information right there. But what about that whole bottle cap thing?
You must be referring to the bottleneck effect. The bottleneck effect is caused by a severe drop in population size, the bottleneck. Few survivors become isolated from their original larger population. Because of this, certain alleles may be overrepresented. Therefore, the allelic frequencies in the population may change, known as genetic drift. The select alleles represented within the group of survivors cause this change. Other alleles may even be cut out altogether. By definition, genetic drift is when chance events cause allelic frequencies to fluctuate unpredictably from generation to generation, especially in small populations.
Ooh, wow. Never knew there were so many neat facts about evolution that Dr. Strunk-Fugen thought. Shh, be very quiet. This is a very rare and beautiful thing. You see the male peacock here is spreading his feathers, attempting to attract a mate and get all up on that peacock booty. See, he spreads his feathers and ruffles them to attract the female peacock. Let's get a watch, come on. Wow, that's pretty swell. This here is one of my favorite exhibits, the monkeys. Over the break, Dr. Gorfenbach and Schrugen taught me all the neat things there are to know about populations, microevolution, gene pools and whatnot. My brain is just swimming with information right now. But first, let's go see some monkeys. Come on, let's go! You see, monkeys can't get jiggy with other monkeys. You see, because they're different populations and species, they can't slide into each other's DMs. You see, a population is a group of individuals of the same species that live in the same area at a specific time and can interbreed, producing fertile offspring. The gene pool of the population is the genetic makeup, which consists of all the alleles for all the loci in all the individuals of this beautiful, wondrous population. Evolution or change in allelic frequencies in a population over generations on this small, actually smallest scale, is known as microevolution. tell us.
When consumers refer to that Netflix channel they have on their post.
In here, the brilliant Dr. Tortoise.
It's Tastrid.
Whatever. So, Dr. Tastrid, can you please tell everyone what you're supposed to tell us about all the things?
You mean the Hardy-Weinberg principle?
Yes.
The gene pool population that is not evolving is called the Hardy-Weinberg equilibrium. It states that the alleles and frequency of alleles and genotypes in a population will remain constant from generation to generation if only some factors are at work. This would result in a stop of all evolution, including microevolution. A gene pool in this scenario is said to be in the Hardy-Weinberg equilibrium.
Man, well, with as hot as it is out here, I'd sure like to go swimming in this gene pool.
Hardy-Weinberg will not occur if one of the 5 conditions of the Hardy-Weinberg equilibrium is not met. If the 5 postulates are not met, evolution will be occurring in the given population. The first condition is there must be no mutations. A mutation is a change in the nucleotide sequence of an organism's DNA. The alleles can be altered by deleting or duplicating entire genes which cause modifications in the gene pool. In addition, there must be random mating to allow for the random mixing of gametes.
Oh boy, deleting alleles? That sounds absolutely unbearable.
Yes, absolutely unbearable.
Unbearable.
In addition, no natural selection can occur. Their survival and reproductive success of individuals carrying different genotypes can alter allele frequencies. Another condition is an extremely large population size. If the population is too small, there is a greater chance for allelic frequencies to fluctuate from one generation to the next. The last postulate is no gene flow. By moving alleles in and out of populations, the allelic frequencies can be altered by gene flow.
Oh, wow, shh, be very quiet. I'm startled. This here's a very rare specimen. This is the West African horned viper of the Zimbabwe. You can tell it's one of those because of the way it is. Thanks for joining us on another exciting adventure of Nick's Nature Time. I got to get back to the research lab, so I got a jet. See you next time.
Go.
Welcome to my laboratory. In this next bit, we're going to delve deeper into the Hardy-Weinberg equilibrium by showing you an equation to give you an example of this mystifying thing.
The equation for the Hardy-Weinberg equilibrium is as follows. P squared plus 2pq plus q squared equals 1, and p plus q equals 1. This is used to test whether evolution is occurring in a population or not, where p equals the frequency of the dominant allele, and q equals the frequency of the recessive allele in a population. P squared equals the percentage of homozygous dominant individuals, and q squared equals the percentage of homozygous recessive individuals. And 2pq stands for the percentage of heterozygous individuals.
Yeah. Is your head sure swimming with information as much as mine is? Whew!
Let's just say the color brown is dominant over the color white in a population of butterflies.
Man, all this excitement sure giving me butterflies.
That's wonderful, Nick. 40% of the butterflies in this given population are white. With this information, we must calculate the q, p, p squared, q squared, and 2pq values in this population. Given that white is recessive, we already know the percentage of recessive individuals, the q squared value, which is 0.40, taken right from the 40%. To find q, we must take the square root of 0.40, or 40%, which would be 0.632. Now that we know what q equals, we can find p by subtracting our q value from 1, using this equation, which would equal 0.368. This p-value can be squared to find the percentage of homozygous dominant individuals in the population, which equals 0.135. The percentage of homozygous individuals is found by multiplying 2 by p by q, which would equal 0.465.
Well, that concludes this episode of Nick's Nature Time. Hope you had as much fun as I did. Well, off to my next adventure. Yeehaw! So, as I'm sure a lot of you know, Lauren Tostred is deathly afraid of birds. So today, she's going to overcome her fear, and we're going to send her into the little bird exhibit we got over here. And she's going in alone. Alright, let's go, Lauren. Don't be afraid. Don't be afraid. Lauren, slow down. Lauren, this duck wants to talk to you. Lauren. She sprinted through that. Just sprinted.